Okay, good evening, everyone. I'm today's presenter, Huiyi Wang, from R&D department in GemPharmatech. Today, I'm going to share the topic of the challenges and strategies of NK cell-related IO therapy in preclinical drug evaluation. As we all know, NK-related therapies are receiving increasing attention. Antibody-type drugs can kill tumor cells by exerting ADCC effect on NK cells. To this end, bispecific, trispecific, and even tetraspecific killer cell engagers are being devised to improve NK cell recognition of tumor cells. Circumventing tumor immune escape and efficiently tagging NK cells to tumors. Moreover, the exciting technique of chimeric antigen receptor engineered NK cells, which is CAR-NK cells, offers unique opportunities to create CAR-NK with multiple specificities, unknown the experience gained with CAR T cells, with potentially less adverse effects. Because of this, NK cells has unique advantages. So NK-related therapeutics marketing is growing at a high speed every year. According to some estimates, the NK-related treatment market will reach 5,096 million by 2026. As we say, NK cells is becoming a rising star in cancer immunotherapies. And this picture shows the process of NK development. In humans, CLP develops the ILC restricted CILCP, which in turn give rise to NK restricted NK progenitors. Both T-beta and AMORs are required for further differentiation into functional NK cells. The expression of the CD156 allows the distinguishing of two subsets of the NK cells, the CD156 bright and the CD156 dim NK cells. The CD156 dim NK cells are cytotoxic, and both subsets are able to produce cytokines like the interferon gamma and TNF-alpha. Two activating surface receptors, NKp46 and NKp18, are expressed by both subsets of NK cells. It has been proposed that CD156 bright NK cells differentiated into the CD156 dim NK cells with the acquisition of CD16 and PEN5 and CD157 expression. Comparing to other immune cells, NK cells have some key features, like NK cells are identified by CD3 active and CD156 positive markers. NK cells makes up about 5 to 15% of human peripheral blood and 2 to 5% of neurospinocytes. And the NK cells has no faster cytotoxicity over injured cells, virus-infected cells, and NK cells express the surface markers like the Nkc16 and the CD157 in human and NK1.1 or NK1.2 in mice. Unlike B cell and T cells, NK cells don't express TCR or B cell. And NK cells is very special. It functions in both the innate and adaptive immune systems. So NK cells play a very important role in tumor immunity. And how do NK cells kill tumor cells? According to current paper reports, NK cells kill tumor cells mainly through 5 ways. The first one is direct cytotoxicity. NK cells are the first responders of the immune system and can directly recognize and analyze tumor cells. Activating receptors on NK cells recognize ligands that are mostly expressed on compromised cells, while inhibitory receptors bind to self-ligands that mark healthy, normal cells. Second is the ADCC effect. NK cells can express the CD16 Fc gamma receptor 3A receptor that binds antibodies and triggers the antibody-dependent cellular cytotoxicity. This response contributes to the efficacy of many of the antibody-based cancer therapeutics, like the Herceptin and the rituximab. Third way is that the T-cell recruitment. NK cells, when activated, will release the cytokines such as the TNF-alpha, interferon gamma, that can recruit other immune cells and heat up the tumor microenvironments for immunotherapy. And the first is that intratumor NK cells produce that CCL5 XL1 free 3 ligand. And the setting of real intratumoral stimulatory dendritic cells can activate the adaptive immune response. And the fifth is that exosome release. NK cells can release exosomes with cytotoxic activity and can contain effector microRNA, cytokines, and display NK cell surface receptors. As we mentioned in the last slide, most NK cells are cytotoxic. They produce lytic granules containing a series of molecules, like perforin and glycamase, that can induce cell death in stressed cells. The regulation of the NK cell effector function is ensured by a dynamic balance between inhibitory and activating cell surface receptors. These inhibitory receptors, such as the inhibitory exosomes of KIRs in human and the CD94 and KG2A heterodimers in both human and mice, will recognize the various forms of MHC1 molecules. Therefore, the decrease or absence of MHC1 molecules on target cells will reduce the stress of inhibitory signals and these signals will deliver it to the NK cells, and then will promote the NK cell activation. The NK cell activation also results from the inhibitory signal, from the engagement of activating receptors, such as the activating exosome of KIR and LY49. The signaling lymphocyte activating molecules like NKG2D and other receptors. So NK cells can also mediate antibody-dependent cell-mediated cytotoxicity upon engagement of Fc gamma 3A by target cells coated with antibodies. There are about 3 mechanisms of NK cell inhibition: downregulation of activating receptors on NK cells due to the shedding of ligands such as the CD155, and on tumor cells, which impacts the activation of activating receptors and the release of suppressor cytokines such as the IL-6, TGF-beta, contribute the formation of the immunosuppressive tumor microenvironment. There are some suppressive immune cells such as the tumor-associated macrophages and the Tregs, dendritic cells, MDSC, and the fibroblasts. All these cells will suppress NK cells, via the suppressive the cytokines such as TGF-beta, L10, L1-beta, and so on. There are, as the Nkcr is a very attractive, effector cells. Many biotechs and big pharmas has developed the drugs targeting inhibitory receptor on NK cells. And nivolumab is a fully human monoclonal antibody against the KIR with potential anti-neoplastic activity. Once administration, nivolumab binds to KIR, thereby preventing the binding of KIR ligands to KIR on natural killer cells by blocking these inhibitory receptors. NK cells become activated and attack cancer cells, leading to the tumor cell death. And KIR is a member of the immunoglobulin superfamily. It is expressed on the surface of NK cells. The other antibody is targeting the NKG2A, which is the molnupiravir. And this antibody is a potentially first-in-class immune checkpoint inhibitor targeting NKG2A receptors, expressed on the tumor-infiltrating cytotoxic CD8 T cells and NK cells. NKG2A is not inhibitory checkpoint receptor for HLA-E. By expressing HLA-E, cancer cells can protect themselves from killing by NKG2A positive immune cells. And HLA-E is frequently overexpressed on cancer cells. And on cancer cells of many solid tumor cells and some hematological malignancies. And this anti-NKG2A mono antibody may re-establish a broad anti-tumor response mediated by NK and T cells and could enhance the cytotoxic potential of other therapeutic antibodies. In addition to the mono antibodies, there are also some newly developed bispecific and trispecific antibodies targeting NK cells that are under development. A bispecific antigen killer engagers, which is called bicos, are small molecules that engage two sites, linking the activating receptor CD16 on the NK cell to a tumor antigen resulting in ADCC. And the trispecific killer cell engagers are a derivative of bicos that also include IL-15 to further enhance NK cell activity. Checkers promote NK cell survival and proliferation to solve the persistence of NK cell, the short persistence of NK cells problem. In addition to inducing the ADCC, also there are the 2019 cell reported that there is a new generation of trifractional NK cell engagers consisting of mono antibody fragments targeting the activating NK cell receptor NKp46 together with tumor antigen and the Fc fragment to promote antibody-dependent cell-mediated cytotoxicity via the activating receptor CD16 expressed on NK cells. And this antibody were very effective against several tumor types in vitro and no off-target cytotoxicity. In vivo, Nkcrs were more potent than intact the anti-tumor antigen mono antibodies, such as the anti-CD20, monoclonal antibody, rituximab, and FC engineered anti-CD20 monoclonal antibodies. These results support the clinical development of Nkcrs for next generation of cancer immunotherapies. Various approaches to therapies with the adoptive transfer of NK cells. NK cells can be obtained from either the patient or from a donor. In autologous transfer, NK cells from the patient are activated and expanded in vitro in the presence of cytokines. The combination of L12, L15, and L18 might generate NK cells that are more functional and have memory properties. The expanded and activated NK cells are then transferred back into the patients, who generally receive cytokine administration like the L2 to sustain the expansion and function of the infused NK cells. Although autologous NK cells might recognize activating signals such as stress molecules on cancer cells, their anti-tumor activity is limited by the inhibitory signal transmitted by self-HLA molecules. To solve this problem, the scientists developed the allogenic NK cell transfer. The NK cell can be obtained from the HLA-matched or haploidentical donors. And the NK cells are expanded through this process similar to the used for the autologous transfer. But T cells should be removed. T cells should be removed to avoid the GVHD occurrence. In this setting, the best responses are obtained when haploidentical donors don't express KRs that recognize the patient's HLA molecules, because donor NK cells don't receive the inhibitory signal from the patient's cancer cells. And the CAR can be engineered in autologous or allogenic NK cells, or in NK cell nets such as Nkcr. CAR engineering endorses NK cells with the antigen specificity. The binding of a CAR to the tumor antigen delivers a potent activating signal that triggers NK cell cytotoxicity, which results in the elimination of the cancer cells. Several strategies have been developed to exploit NK cells' anti-tumor properties. First, cytokines. L2 and L15 is well known to boost NK cell activities are being tested, but the immune responses, adverse immune responses is a very serious safety issue. And second is the activated NK cells. NK cells from cell donors can be stimulated with the L2 and L15 in vitro prior to infusion into the bloodstream of cancer patients. This strategy can take advantage of the mismatch between the donor KR and the patient's human HLA to reduce the inhibition of NK cell function and promote their anti-tumor activities. And the third is that genetically modified NK cells, autologous or HLA mismatched. Chimeric antigen receptor NK cells, genetically modified to express the anti-CD19 or anti-CD20 receptors against the B-cell malignancies. And the L2 or L15 could enhance the NK cells in survival. And first is that monoclonal antibodies. This, as we introduced in the last slides, the therapeutic antibodies tagging tumor-associated antigens, such as the rituximab or cetuximab, recognizing the CD20 or the HFR respectively, can induce the ADCC by NK cells. The antibodies directed against inhibitory molecules, such as molnupiravir, recognizing the NKG2A, we introduced before, potentiate NK cell responses. And NK cell engagers, multifunctional antibodies tagging tumor antigens such as CD19, CD20, or HFR, while bring NKB146 and CD16 will bring tumor cells and NK cells together, therefore triggering tumor cell destruction and cytokine secretion by NK cells. There are roughly two types of mouse models widely used to evaluate NK cell-related therapy. The first is genetically engineered mouse models, and the second is the NCG-hIL15 humanized immune reconstituted mouse models. In NK-related GMM mouse models, we replaced the mouse gene with the corresponding human genes. GemPharmatech has developed 20 plus NK-specific humanized mouse models for testing different NK-associated test articles' efficacy. Here is the list of GemPharmatech's humanized mouse models. The red means past the mRNA verification, and the black means the under development. There is still some challenges faced by NK therapies. In the HRC NCG mouse models, NK progenitors can't or rarely develop into the immature NK cells because without the support of the IL-15. And cytokine IL-15 is very critical to the development and the persistence of NK cells. But most IL-15 poorly support the human NK cells due to the low affinity to human IL-15 receptors. So human IL-15 needs to be inserted or injected into the NCG mouse models to help the NK cell development. To solve this problem, GemPharmatech has developed the NCG-hIL15 humanized mouse models. One of the limitations of adoptive NK cell therapy is the lack of the in-vivo persistence of the infused cells in the absence of cytokine support. To solve the limited persistence of NK cells, you have two options. The option one is the repetitive NK cell infusion, which is very expensive. And the option two is the addition of cytokines of IL-2 or IL-15. The administration of cytokines has been shown to enhance the proliferation and persistence of adoptive infused NK cells. However, it can also induce some cytotoxicities, as well as the expansion of other immune subsets like the Tregs. Hence, so several groups have reported that has reported encouraging results by engineering NK cells with transgene encoding for cytokines that are either expressed on the membrane or secreted constitutively. In one study, NK92 cell lines or primary NK cells transduced with retroviral vectors expressing L2 or L15 showed enhanced proliferation and persistence in tumor-bearing mice. Our group has shown that incorporation the L15 transgene into the CAR construct enhance NK proliferation and in vivo persistence, and with improved anti-tumor activity. In NCG-hIL15 mouse models, there is no endogenous release of human L15, which could support the L15 persistence in the periphery and inhibit the tumor metastasis. From the data in the published paper. And we have compiled the mouse L15 and human L15 MR expression level between the NCG and NCG L15 humanized mouse models, and we found that whatever in the heart, liver, spleen, lung, kidney, intestine, stomach, muscle, ovary, marrow, blood, brain, in all these tissues, we found a comparable expression level between the mouse L15 expression in NCG and the human L15 in NCG-hIL15 humanized mouse models. And in ELISA assay, and we found there is a relatively there is the human L15 expression in the plasma in the peripheral blood. GemPharmatech has developed 3 methods to reconstitute human NK cells, and the most classic is engrafting the CD34 positive HSC cells into the irradiated NCG-hIL15 mouse models, which could reconstitute both T, B, NK cells. And the second newly developed method is engrafting screened Huiyi cells or the NK cells purified from the PBMC cells into the NCG-hIL15 mouse models, which also could reconstitute the NK cells. But in the PBMC engrafted NCG-hIL15 mouse models, there is a reconstitution of T cells. But in the Huiyi or engrafting NCG-hIL15 mouse models, there is only NK cell reconstitution in peripheral blood. But in PBMC mouse models, the window period is relatively shorter, and in human NK mouse models, the window period is over 12 weeks. And from the purified NK cells from PBMC or engrafted into NCG mice or NCG-hIL15 mouse models. And in the NCG mouse models, the proportion of NK cells in peripheral blood suddenly disappeared from the second week. While in the NCG human IL-15 mouse models, the NK cells could continue to proliferate until the 10th week. Mice are monitored for body mass changes every week after PBNK infusion, and survival rates were collected. The the body weight and the survival curve is relatively stable in these mouse models. And the NKs were tested in NCG and NCG-hIL15 for preclinical efficacy evaluation. And the mere PACA2 tumor cell lines were inoculated. The frequency of administration was once a week. And in the NCG-hIL15 humanized mouse models, we can see that CAR-NK treatments can significantly increase the tumor size, and the number of NK cells in the peripheral blood is relatively high comparing to the NCG mouse models. Here is the Huiyi NCG-hIL15 mouse models. Comparing to the Huiyi NCG group, human IL-15 insertion wouldn't shorten the lifespan after HRS engraftment. Also, the body weight is stably growing post-engraftment, which means the HRS transplantation into the NCG-hIL15 mice didn't induce the undesirable toxicities. Comparing to the Huiyi NCG-hIL15 mouse model, the Huiyi NCG-hIL15 has a higher human CD15-5 positive nucleocytes reconstitution level. The reconstitution of NK cells doesn't affect the T cell proliferation. It ignores the B cell proliferation, but the B cells in the Huiyi NCG-hIL15 mouse models is the immature B cells. It lacks the ability of class switch, so it wouldn't influence the total immune system. And obviously, it promote the NK cell reconstitution. At the 13th week post-engraftment, functional markers on NK cell membrane in the peripheral blood of Huiyi NCG-hIL15 mice were detected, and functional markers such as KR3DL and NKG2A were detected, and NKG2D were detected on NK cells. Comparing to the and the reconstituted NK cells from the spleen were separated and purified by commercial kits. For cytotoxicity experiment, NK cells and K562 cells were mixed with different effector and target ratio. From the data, we can know that NK cells from the Huiyi NCG-hIL15 mouse models could directly cure the K562 tumor cells. And in vitro ADCC study, rituximab and Raj were used. Rituximab is an anti-CD20 antibody, and many depends on the ADCC function. By concature with NK cells with Raj and rituximab, we found NK cells could kill Raj cells by ADCC function. And then the in vivo study was further proved that rituximab could inhibit the tumor growth in Huiyi-hIL15 humanized mouse models. And this data showed that the Huiyi NCG-hIL15 mice could generate functional NK cells. This mouse model could be used for immunotherapy, which targets NK cells. Also, we have conducted the NKG2A and efficacy study. The administration of NKG2A could obviously inhibit the NCH1975, the tumor cells in tumor growth in the HUHS and NCG-hIL15 mouse models. And this data is collaborated with the third party. And at the study endpoint, we collected the mouse tumors and conducted tears analysis, and found that the administration of NKG2A will downregulate the NKG2A expression on NK cells, which means that NKG2A monoantibodies will inhibit the tumor growth by blocking NKG2A binding to the HLA-E. And here is the Huiyi NCG-hIL15 and Huiyi NCG-hIL15 two mouse models. The T cell reconstituted only in Huiyi NCG mouse models, but NK cell reconstituted both in the Huiyi NCG-hIL15 and Huiyi NCG-hIL15 two mouse models from the cell counts data. I also conducted another study. We purified NK cells from different donors to comparing these donors to compare if all the PBMC donors could support the reconstitution of NK cells in NCG-hIL15 mouse models. The human NK from different donor sources were inoculated into NCG-hIL15 mice with a dose of 2E6 to test the NK cells engraftment efficacy. As we can see, donor 1, donor 3, and donor 4 exhibit higher capacity to reconstitute human NK cells, but donor 5 is weak in NK reconstitution, which means donor selection is very important and necessary. And the GemPharmatech will offer the tumor selection. Will select the PBMC donor by in-vivo reconstitution test before the offering service to our customers. Here is our take-home tips. Nkcr is a rising star in cancer immunotherapy. Modulating NK cell function can be achieved in a variety of ways. Anti-inhibitory agents like anti-NKG2A. The presence of IL-15 is important for NK cell development and function in vivo. NCG-hIL15 is a good model for preclinical agent evaluation that requires NK cell presence and function. NCG-hIL15 is a good model for CAR-NK preclinical evaluation. There were several major limitations inherent in humanized mouse models, in part due to the differences between mice and humans in the growth factors and cytokines required hematopoietic and immune system development. Additional limitations include deficiencies in the development of lymphonodes, poorly organized lymphoid architecture, and impaired affinity maturation and class range of antibodies following immunization in the humanized mouse models. In humanized mouse. Besides NCG and NCG-hIL15, GemPharmatech has developed various next-generation NCG mice to support specific immune studies. To support the lymphonodes development, we developed NCG mouse TSLP. To develop the myeloid, macrophage, and D cells, we developed the NCG SGM3 mouse models, and also the NCG-3 mouse models to support the DC development, and NCG-X for the red blood cell development, and others. And thanks for your attention. Yes, that's today's webinar. Okay, that's all. Thanks for your attention. There is a question that asking what are characteristics needed in mouse models used to evaluate CAR-NK therapy. Actually, for the limitation for CAR-NK therapy is that the persistence of CAR-NK in the mouse models. So to solve this problem, I think different biotechs may have different this problem existing in mouse models also existing in humans. To solve this problem, different companies have different strategies. Firstly, you could for the preclinical evaluation, we usually will use the human IL-15 injection compound human IL-15 injection into the mouse models, or directly using the NCG-hIL15 mouse to conduct the CAR-NK therapy, which has a better efficacy study result. Yes. Okay. CAR-NK therapies targeting CD22, CD19, BCMA, CD13. Oh yes, we have these mouse models, and we have enormous the different CDX tumor cell lines in our in our library to conduct this CAR-NK therapies. And there is a question that can you say something about toxicity assessment of such drugs? I'm sorry, we didn't do the toxicity assessment in our mouse models, so I can't give you a professional advice in this webinar. There would be GVHD when put human immune cells into mice. How to pronounce the evaluation window? It's an open question, I think. To pronounce the evaluation window, we have several methods. Firstly, I think if you concentrate on NK cells, we will suggest to use the Huiyi NCG mouse models or Huiyi NCG-hIL15 mouse models. It has a no window period, and but as I said in before, during the Constitution needs a long time to constitute the NK and T cells. To solve this problem, we also construct the PBM, the NK cell, the human NK cell in graft transplanting. Transplant into the NCG-hIL15 mouse models to solve this problem. Yes. Okay, thanks for your questions. Have a good night. Thanks.